our Honorable Vice Chancellor, Sir, Professor Soikat Moitro, inaugurate this program. Also with us, Dr. Parthopratim Lahiri, Registrar of our university, Dr. Otri Bhaumik, the Finance Officer of our university. He also, Professor Raja Banerjee, who is the coordinator of this STTP training program. This would be counted as half FDP and certificate would be awarded accordingly. The program would be 6 days long and every day there would be 2 sessions of at least 2 hours each. Details of the program has already been circulated and known to the participants. So before we go for the inauguration part, I request Professor Raja Banerjee to say a brief introduction and planning regarding this STTP. Professor Banerjee, please.
Good morning to you all, Honorable Vice Chancellor Sir, or the Register Sir, our dignitaries, and my dear participants, along with this Mr. Shohom, my beloved student, and Prem Kumar. With all, with due respect to all, I just want to mention that biology today is not an individual subject, a subject of individual identity. It is nowadays basically an interdisciplinary subject where the knowledge of chemistry, physics, mathematics, and computer science is essential. And considering this aspect, in order to upgrade and develop, and what is the perspective in today's respect, as well as what is the future demand, considering this thing under the able guidance of AICT and our honorable vice chancellor, we are planning and we are now pursuing the FDP program, which already our controllers are, that is Mr. Dr. Datto has told you, it is an FDP program, faculty development program, where we are giving focuses, impetus on the basic needs of more current as well as the future biology. And for this, we have talked to several persons who are not only persons who are basically the eminent scientists in each and every corner of the biology. And I am very much thankful to all of them, all these eminent reputed scientists of the different part of the countries. They gave their kind consent and they will act as a resource persons. And actually, not only the faculty of a particular subject, not only a particular discipline, the faculty from each and every discipline, even the industry, the ex CEO of the AstraZeneca will come and he will tell us how the academy and industry handshake each other, and from the knowledge of the academic, how industry is giving the products. We have with us a very eminent, reputed scientist, Professor Satyajit Roth, who was basically a physicist and then became a doctor and now become a biologist and one of the pioneer immunologists of the country. So we have faculty from ISR Pune, IISc Bangalore, cultivation of science and etc. So we will see in near future how they will enrich you and through which our future generation will be enriched, who will be the torch bearer of our country. So one thing I want to clear, I want to mention, I got a phone call from one or two persons, same name in session 1 and session 2. It is not the same repetition. Basically, these are the continuation. In the first part, morning part, the session will start with a recapitulation of the knowledge and how you can build it. And in the second part, the same will be continued with the future aspect. So there is nothing to say a repetition of anything. So it will be progressive way. So with this, I hope this endeavor will be successful and our effort will be actually encouraging you, enriching you, and through which that will actually enlighten our future part of the country. With this, may I request Dr. Dotto to request our Honorable Vice Chancellor to inaugurate the program through his inaugural speech.
Thank you, Professor Banerjee. Now I request our Honorable Vice Chancellor, sir, Professor Soikat Banerjee, to inaugurate the program with his inaugural speech. Sir, please.
Soikat Moitro.
Sorry.
Good morning to all the dignitaries present over here, my colleagues, Dr. Lahiri, Dr. Datta, Dr. Bhumik, Professor Raja Banerjee, and other members from our university and all the participants. This particular endeavor we have started recently with the support from AICT in the form of a short-term training program, and which, as described by Dr. Datta, that it may be considered as a half FDP program, half FDP equivalent, or all. So it is a good thing that all the participants, they will get credit for attending this particular training program. Now, regarding this today's topic, and the topic based on which this short-term training program will continue for the coming 5-6 days, is biology for mankind. It appears to be a broad-based, these titles, and it may cover maybe different aspects of biology. However, this importance of this biological science, this in the coming decade, in coming, this times, is enormous. Biological science and information science, these two things will be dominating the entire world as far as the technological development of is concerned. Particularly in country like India, where these two things are gaining this Gurgantine importance, because the fact is this: here we have this tremendous this quality of biodiversity till now, which we have not this leveraged this benefit to the best possible extent. There are many untapped things in different parts of this country. And in near future, there's many different areas, be it healthcare, be it agricultural sectors, be it these food sectors, everywhere. The contributions and interventions of biological science and information science will be stupendous. And already we have seen that researchers are taking up many research activities based on this particular primary areas. Based on, so this is one of the changing scenario worldwide, that most of these researchers, they're focusing these researches in this particular period, which are going to be of utmost benefit to human race: information science and biological science. And in what we see, what we can this foresee right now, that if we empower our research community or next generation with the right set of skill, right set of this knowledge, and of course right set of attitude, then we can do a lot of things to change the entire this academic scenario in terms of these economic propositions. Change. We can support this different micro entrepreneurships, knowledge-based entrepreneurships, to change the economical the scenario of this country, providing the scope of engagement and employment a large number of people. And of course, we can open up different frontiers of new areas, new research areas, new this knowledge areas by doing this. And here we need to take up a concerted effort in that direction. We are very happy ICT is supporting these sort of endeavors. Our these senior professors, they are taking keen interest in organizing these types of training programs, inviting personnel from resource persons from different universities, different institutes. This will require to a significant, formidable extent, because this is the time for developing effective networking and collaboration. I repeat this phrase at many occasions. And this platform, this brings this opportunity of effective networking and collaborations with many researchers across this country and maybe of the other countries as well. So with this, I wish a grand success for these short-term training programs. I am thankful to all these resource persons who are present over here and who will be delivering lectures in the sharing their knowledge, sharing their these ideas with our participants in the coming few days. And my sincerest thanks to Dr. Subhashish Datta, Professor Raja Banerjee, for taking up this particular task in making this successful. And my thanks to AICT also for supporting these endeavors. And I believe our participants, our research community, our learning community, they will be immensely benefited out of this exercise. Thank you all.
Thank you, sir.
Thank you, sir. And Dr. Dattu, I am very happy to see that our AICT director, Colonel Venkot, has joined.
Yes,
So,
yes.
if you kindly.
Yeah. We are happy to see that Colonel Venkat, as always, supporting us, has joined this program. He was at the inauguration session of the first program as well. So today is the second one we are inaugurating. And this is for all the information for all participants that in each Monday we'll start a separate program in STTP this month. So probably every time we'll get the support of participation from the Colonel Venkat as well. Colonel Venkat, I'll request you to say a few words to the party gathering in this occasion.
Thank you, Datta sir. It's always a pleasure to be coming and speaking. And with the regards to the Honorable Vice Chancellor sir, and to the university and all to faculty members, it's a pleasure to be coming on your online platform in which you are starting now. I recollect coming on the previous one, and as told already speaking, Subhashish sir did mention that there's going to be a weekly kind of an activity and program, so that's wonderful. With every time you're connecting with a whole lot of faculty members, and when every time you're connecting, you're coming out with a new topic. So there is a new set of faculty members connecting. There are new set of people who are getting associated with you. Quite obviously, beyond the normal activities of broadening the horizon of many more faculty members who are getting associated with you, you are also establishing the touch and connect across the length and breadth of the country, and to make sure that people who actually wish to gain knowledge come forward on your platforms and are going back a little bit more learned than what they were already. So suffice to say the least, that every time you do an activity, you're bringing about the latest, you're bringing about the highlights, you're bringing about some futuristic activities. And today when I see your topic, it's biology for mankind. Oh, this is a very exciting topic, and quite obviously, I believe you're going to come out with some fascinating conclusions at the end. You're going to come out with some very relevant and pertinent points at the end. The takeaways are going to be very good to the effect that people are going to be benefiting from this topic, and why not? What we are seeing happening all around us today and in the last 1 year is nothing but a culmination of our lack of application involving the biology, involving various other subjects. Even though in engineering we have a subject called as biotech engineering, or you might be having biomedical, but our understanding of the subject is not much. In fact, I would rather also give you a small indication. AICT has just about written a book on professional education on behest of the Government of India. That professional education chapter is as part of the National Education Policy as to how best the agriculture, medical, health, and legal aspects should also be there to each and every individual and graduate who is now passing out of any higher education, not only the higher technical, of any graduate degree that he takes. So this particular subject also brings about a little bit more understanding of the subject to a lot many more people who are directly not connected with biology. So compliments to Subhashish sir and your university for selecting very, very pertinent topics and a varied topics spreading across the length and breadth of all your disciplines and streams. And it's a pleasure and honor to be associated with you. Be rest assured, AICT will always be connected. Be rest assured, we are looking forward to all these activities. And then we are also looking forward to go beyond this into many more associations with your university. The Honorable Vice Chancellor, sir, with due regards to you and all faculty members, it's a pleasure to be coming and speaking. My apologies for joining a little late, and wishing you all the best in your endeavors. And I would request, please continue doing the good job. Your last STTP or STC program, which we call, had wonderful reviews. Please be rest assured, we take direct reviews also. That's also our mandate. We take direct from the participants. It was a wonderful review that we got from your previous program. Hoping and expecting that you're going to be crossing those expectations also in this particular time. With deep regards and wishing you all the best and best wishes. Jai Hind.
Thank you, Colonel Venkat. It's always good to listen to you, and you have been always supporting, and you are not only supporting in this STTP program, but also there are many other programs like translation of books, and there are so many other programs you are supporting also for staff development program. So we are happy to get the support and work together with you. I can ensure you the a kind of combined report would be submitted to the AICT after completion of this program with the feedbacks of the participants and the achievements we did. And we are happy to know that the new initiatives of AICT, the book you are publishing in respect of the new scenario. So we'll definitely, once we get those copies, we'll definitely share with our teaching community and student community. Thank you. Thank you, Colonel Venkat. Now, I'd like to request Registrar Dr. Parthopratim Lahiri to speak the gathering.
Good morning to all. Honorable Vice Chancellor, Professor Sholkat Moitra, Honorable Director of AICT, Colonel B. Venkat, Professor Raja Banerjee, the Director, School of Biological Sciences, my colleague, Dr. Subhashish Dotto, Controller of Examination, and also the coordinator of the entire AICT program, and also my colleague, Dr. Otri Bhomik, the Finance Officer of the University. And obviously, dear participants, it's really a very nice occasion to meet you on such a platform where a 6-day STTP program, a training program, a biology for mankind, will be delivered to you. I hope that the program will be a grand success. As mentioned by Professor Banerjee, this is an interdisciplinary or cross-disciplinary kind of program, and attending this kind of program, you will be able to understand that at a certain points, all the subject mates like biology, computer science, mathematics, physics, chemistry, they all meet at a certain point, and that point is the point from where we can do good for the mankind. So obviously, the training on this kind, the training that you will be receiving, or the lectures that you will be hearing from renowned persons in the field or from the adjoining fields, will be very helpful and beneficial to you. And after 6 days program, when you go out, it will not only help you to achieve your CAS or professional career development, but also to help you academically. And hopefully you will find new avenue to work further in the future time. So I once again hope a grand success. I also welcome all the participants on behalf of the university. I would also like to thank AICT for supporting this kind of training program. And it's always good that this kind of refresher or orientation type of program is given to the university for refreshing the knowledge of the teachers or upgrading the knowledge of the faculty members. I once again hoping a grand success, of this program, and hope that in future the further the modified version of this kind of training program will also take place. I thank you all for your patient hearing. Thank you.
Thank you, Dr. Lahiri. I can see Professor Sutirthi De, our first resource person of the program, which will be commenced from 11 a.m. has already joined. Sir, and thank you for joining with us. So Next, I'd like to request our finance officer, Dr. Otri Bhaumik, to address the gathering. Dr. Bhaumik. Dr. Bhaumik, you are muted.
Sir, please unmute.
Good morning, everyone. Respected Professor Soikot Moitra, our Honorable Vice Chancellor, sir, Colonel Venkat, Professor Raja Banerjee, our respected Registrar, Dr. Parthopratim Lahiri, and AICT Coordinator, Dr. Shubhashish Datta, respected panelists and my dear participants. It's really a great opportunity and great moment for us to organize such kind of STTP on faculty development in association of the AICT. We are really grateful to AICT for providing us necessary input, necessary resources for holding such kind of very productive event for the faculty members. I hope that this faculty development program is going to be a very productive one, and it will help you to excel in future, and the ultimate beneficiary will be the students at the end of the program. Thank you very much.
Thank you, Dr. Bhaumik. Now, before we go for concluding the session, I'll request Professor Sutir from ISAR Pune to say a few words in the inaugural session. Professor Dey, please.
Yeah, I would really like to thank WBUT and Professor Banerjee for organizing this very nice meeting, and I'm sure that it's going to be a very useful and a cracker of a meeting for the next 6 to 7 days. So that's pretty much all from my side. I hope to say a lot more over the day as the meeting progresses. Thank you.
Thank you, Professor Dey. Now I request Professor Raja Banerjee to give vote of thanks.
So every program, there should be an end. And in this inaugural program, this is the end where we have to give a vote of thanks. And initially, I start with AICT, without whom this program cannot be possible. And actually, once again, I am very much thankful to AICT and the Colonel Venkat for your kind gesture and extending your all kind of support to organize so that we can do our duty at the best level. Next, I must thank to our Honorable Vice Chancellor, under whose able guidance and support and leadership we are organizing one after another program. And actually the vision and mission which he is showing to us, it is our duty to fulfill and to converge so that we can help the country and to a better one. So once again, sir, thank you very much. I am extremely thankful to all the administrative staff support from the university, especially from the registrar sir, the FO sir, and our coordinator of this STTP program, Dr. Shubhashish Datta. Without their support, this cannot be done. Especially, I must mention the name of my student, beloved student Shoham, who during my sudden unfortunate incidents, who wholeheartedly supported me, and I know the attendees, the participants who received mail from Shoham, got the phone call from Shoham, without whom this program cannot be possible. So my best wishes to my beloved student Shoham. Thank you very much for your support at my distress condition. And at the same time, Mr. Prem Kumar, who is working always behind the curtain, I will request at least, if possible, Prem to just open your video and to show people should know who is working our behind. So Prem, thank you so much for this. And also I must thank last but not least all the participants, because without whom this cannot be done. And at the end, I am very, very thankful, grateful in the word to say to all the panelists, the eminent scientists, reputed scientists of the premier institutes of all over the country, as Colonel Venkat mentioned, the length and breadth of the country, from the Pune to Bangalore to Kolkata to all these people are coming, and they are just with one phone call agreed to appear as a resource person. So once again, my heartfelt thanks to all the panelists who take their duty and to make our country, the future country, as a better future. So thank you so much.
Thank you, Professor Raja Banerjee. We are not in time. With this, we are closing our inaugural session. The panelists for the inaugural session will now leave the program, and the resource person and the coordinator will remain be there to continue their next session from 11 a.m. Thank you, everybody.
So
Thanks,
thank you.
Thank you very much.
Sir, we may start the session.
Now, this is just going to 11, and our session starts from 11 a.m. So we have with us today Professor Shutirth De, who got his PhD from JNCSR, Bangalore, and he joined as a faculty ISER Pune. He is a very reputed, renowned biologist and working in the field of ecology, chronobiology, and basically the evolutionary biology. So with this very little 1 or 2 lines introduction, because he doesn't demand any introduction, his work and his publication is basically standing for his introduction. So with this, may I request Professor Shutirth, who just only one phone call agreed in spite of his busy schedule and agreed to act as a resource person. So may I request Professor De to start with your session.
Thank you, Professor Banerjee. I'll take it from here. Prem, are you around?
Yes, sir.
Prem, can you please convert, make sure that all the participants are visible? I would like to have a quick chat with them before we start. Great. Great. May I request the participants to kindly switch on their videos? In these kind of situations, it's nice to see who is it that we are discussing with. So for a brief moment, may I request the participants to turn on their videos?
Dear all participants, please switch on your videos. We are making you in the front. So please switch on your videos. As sir has told that it will be some interactive session. So let us make it interactive, and please switch on your videos. And please make the session very interactive. We hope that you are able to get our message. Yes, yes, ma'am. Yes, sir.
Yes, your host would like to.
Please switch off your mic if required only, then please switch on your mic.
Okay. Okay, I think most people are not in a mood to show their faces. Fine, no problem. No, so before I started, I just wanted to have a quick idea. Milan Kumar Maiti, we are getting a lot of sound from your mic. Would you please mute your mic? Thank you, Milan. Yeah. So before we started, I wanted to have a quick understanding of, what is it that all of you are here for. And I also wanted to discuss with you quickly, given what your expectations are from this program, so that I can tailor my content accordingly. So Professor Banerjee told me that all of you are faculty at various colleges and universities all across India, primarily from Bengal, but mainly from, also from other parts of the country. So would you mind, some of you just telling me quickly about your backgrounds and what is it that you feel, you would like to have from this entire session? Professor Banerjee is here, so I'm sure he will be happy to know what is it that your expectations are. So anyone?
Dr. Kaveri Ganguly, ma'am. Ma'am, you can switch on your mic. Yes, ma'am.
Very good morning
Hi.
To distinguished dignitaries. I am Dr. Kauri Ganguly. I am faculty and head of the department in Department of Biomedical Engineering at JIS College of Engineering, Kalani. I am working in this interdisciplinary stream for last 15 years. My research interest is clinical oncology, cellular physiology and biochemistry. And I have worked on the role of estrogen progesterone on insulin-induced maspin synthesis, which is a anti-breast cancer protein. And now my research interest is having twofold. One is biomedical engineering. There I specially work on the domain of biomaterials, and also I am working in this clinical cancer science. And I expect a huge state-of-the-art and very advanced work which are going on around this domain, which will be very beneficial for society and mankind. I'll take that knowledge from this STTP FDP, from eminent persons and researchers, scientists who are coming as our distinguished speaker here. Thank you, sir.
Thank you, Dr. Ganguly. Anybody else would like to say something?
Sir, Dr. MD Iqbal Qureshi.
Dr. Qureshi, okay.
Dr. Qureshi, sir, please. Sir, switch on your mic. MD Iqbal Qureshi.
I think very good morning to every dignitaries present over here. Myself, am Mohammad Iqbal Qureshi. I'm audible to, I think, everybody.
Yeah, you are audible.
Yeah, actually, I am from Kalyani Government Engineering College, working as an assistant professor for last 11, 12 years, and in the Department of Information Technology. Actually, I am working with the area of image processing, pattern recognition, AI. Now, my mission to join this course is I would also like to add some aspect related with bioinformatics. So I have a keen interest in bioinformatics, and I would also like to add some aspects if I can join image processing with that. I'm trying to get this flavor as well. So I'm also very much thankful to the organizer, especially Raja sir, who have given us this opportunity to join this prestigious course at this prestigious institution. Thank you very much, sir. And to you, sir, also I'm very much thankful. I would like to listen to your valuable comments and lecture to learn this new area for me. Thank you very much, sir.
Thank you, Dr. Qureshi. So this is obviously that we don't have time to talk to all 30 of you, although I would have loved that. So the basic thing I think that is happening is, as Professor Banerjee had told me, that all of you have your own areas of research, and obviously, most of you I don't think are working on evolution, and there is no way I can give you any factual information on your own areas of research. Okay, because I don't work on those things. I work on population ecology and population. And what I'm going to do is I'm going to slightly deviate from what Dr. Ganguly put as her expectations, and instead of talking about very latest stuffs, I'm going to talk about certain things in the context of ecology and evolution, the basics of which I'm pretty confident that everybody over here are well aware of. However, what I'm going to focus on is how that relates to the field of evolutionary biology. And why should I do that, given that most of you don't do evolution? The reason I want to do that is because I would like to show you that whatever area you work with, coming from an evolutionary perspective, might end up having some benefit or the other in terms of how you look at the problem. I mean, end of the day, most of us over here are biologists, right? And somewhere or the other, whether we like it or not, evolution ends up touching whatever thing that we end up doing, whatever problem that we end up investigating, right? So what I'll do is, obviously, I will not completely leave you aside from latest stuffs. I will also present some latest work, but that will come as an aside, and that will come in the context of some of the other topics that I'm talking about. Okay. So the way I was thinking of structuring this whole thing, I wanted to first give an overview of variation in biology. And the reason I want to do that is, we had a very long discussion when this entire program was being set up. And if you look at the major concept that is kind of connecting all the talks, which looks, on the face of it that they're very disparate, but actually there is a connecting thread. And that connecting thread is essentially about the kind of variation that you find in biological systems. Now, after this, most of the stuff will focus on the genetic variation. Okay, what do you do with genetic variation? Why genetic variation is important? Okay, some amount of stuff Professor Banerjee is going to talk about in the context of proteins, but end of the day, everything will be connected from a very genetic perspective. So what I'll do is I'll give you an overview of variation in biology. I'll give you some interesting tidbits about why we think that is changing how we look at evolution, and why that change is something that is going to perhaps impact what many of you do. And then in that context, I'm going to talk about some of the latest stuff that we are excited about. And then there are two talks over here. So one of them will happen in this session, the other will happen in the next session. And in the next session, we'll also have a small hands-on stuff. Since most of you are teachers, I'm sure that even though you're not teaching evolution, I'm sure you are teaching something or the other where you have to grab the attention of students. And I'll try to give you a tool just to see whether you can check out whether that works for your students or not. So let's start with evolution. Rather variation. Okay. Just one thing before I start. So this is supposed to be an interactive session. So please feel free to interrupt me at any point. Okay. I will stop whenever it's 1 o'clock or my talk finishes, whichever is earlier. Okay. So please don't be worried about the fact that if we ask questions, then it goes past 1 o'clock and all life will come to a standstill if it goes beyond 1 o'clock. It will not happen. I'll definitely stop before 1. The other thing is that when I'm going to present my screen, at that point, I can't see the chat box. Okay. Therefore, if you have any questions, if you have any comments, I seriously request you to just unmute yourself and speak it out. Okay. That's the only way I'll be able to hear it. Otherwise, if you just type it, I won't be able to see. And I will also be very happy if your questions and comments by and large come while I'm talking rather than, bunching them right at the end. The reason is that there is a flow to the whole thing. And if something doesn't make sense to you while I'm talking, then, the flow will break and very soon you will be like, what the hell is he talking about and why should I listen? Right? We don't want to go there. So anytime, any question, any queries, any comments, unmute your mic and please say it out. Okay, so let's start off. Can all of you see my screen?
Yes, sir, it is visible, sir.
Visible, na? So now I am in the presentation mode. Okay. So as I said, variation a central concept in biology. Now, why am I calling variation a central concept in biology? Because if you think about it, one of the major distinguishing features, one of the things that stands out in biology is that you have variation everywhere. And one of the unfortunate corollaries of having that variation is that it's very difficult to come up with a theory of biology the way, for example, physicists have done or chemists have been able to do. But at the same time, for those of us who do biology, it is this variation that draws our attention, that makes it interesting for us. And if you think about it closely, the entire process of evolution is essentially how this variation is flowing over time, how it is changing, the patterns of the variation is changing over time. People have conceptualized evolution in those terms. So we'll quickly have a look at various kinds of variation, and we'll talk about how that impacts how we think about biology. So why is variation a big deal in evolution? Where is the variation and how is the variation generated? These are some of the major questions that we are going to ask. So why is evolution a big deal? Why is variation a big deal? We all know that variation is the raw material for all microevolutionary processes, particularly natural selection. If there is no variation, no selection will happen, an organism will not be able to evolve. However, what I said over here is true for what is known as microevolution. This is the kind of evolution that Darwin was talking about in his book. However, if you think about what Darwin said properly, you will realize that Darwin's processes, he was essentially talking about how existing variation gets shaped by selection to form something slightly different. And then again, selection operates on it, and again, something slightly different happens. Okay, the slow change part in evolution. However, if you think about speciation, formation of a new species, something so different that we will automatically say that this is completely new thing, then you can't really get there by small changes, because small changes after all will just increase or decrease the value of something that you are measuring. It will not create a new thing. So for the process of formation of new groups, what you need is novelty. What you need is a new trait. What you need are wings evolving when there were no wings earlier. What you need is, claws evolving when there were no claws earlier. And those kind of things are a very different kind of variation. Okay, they are still coming out from what is there in your genes. They are still coming out from normal physiological processes, but their nature can, in principle, might be very different. And for a long time, people in evolution didn't really recognize that these kind of variations might be important. They thought that if you simply take Darwinian kind of evolution and integrate it over a long time, you will end up getting a new species, no problems. But if you think closely about it, that's not necessarily true. And this is one major thought process, one major line of thinking that has been forcing evolutionary biologists to look at the entire process of biology very differently in the last 10-15 years. The so-called macromutations and how they affect the so-called macroevolution, the formation of new species, new groups. So next question, where is the variation? And the short answer to that is that variation is there in pretty much every single level of biological organization. If you take the level of biomolecules, that is DNA, protein, RNA, you have variation, you have it across cells, you have it across tissues, you have it pretty much everywhere. Now, this discussion and this entire workshop actually is going to concentrate primarily on this part, the biomolecules part. Now, how you look at variation, that is a very cute problem, and you look at it in different ways depending on what is it that you want to do, what is the problem that you want to solve, what is the question that you want to answer. So I'm going to talk about various ways of looking at variation over this entire talk. So for example, this is, at the most basic level that one can see, this is in terms of skin color variation, okay, the observable variation. So what you are seeing over here is one single family, a extended South African family, where you can find this person who has a very dark skin color, and you can also find this person, or let's say this person. These people, they are having much fairer skin colors, and you have pretty much every skin tone in between. So where is this variation coming from? So if you go into the causal part of it, this variation comes from three major sources. So if you take babies right at birth, you can see that there is already a lot of variation in terms of their skin color. So this is what is happening due to the inherent genes that are there, the so-called genetic variation. Now, once these babies start growing up and once they are exposed to the sun, you know that tanning happens, and there is also a difference in terms of their skin color depending on how much sun exposure they have seen, what kind of chemicals they've been putting on their skin, and so on and so forth. So this is what is environmental variation. And then you have a third kind of variation, which is the most interesting one, where there is an interaction, a statistical interaction between the two. So some genotypes, depending on what environment they get exposed to, form certain phenotypes, and other genotypes being exposed to the same environment form a different phenotype. Okay. So to put it all together, so these are the three things: genetic variation, which is due to differences in the DNA; environmental variation, which is due to external factors that end up affecting how much protein is made or how the protein is working, and so on. And what you have is also the G cross E interaction, where the genotype by environment interaction, where different genotypes behave differently based on which environment they find themselves in. So when it comes to genetic variation, we have a massive amount of information. I mean, most of you probably specialize in some form of this or the other. How do changes in the genetic sequence change the way the phenotype is expressed? And classically, this is what was thought to be the primary source of evolutionarily relevant variation, because this is what is inherited. I mean, as we all know, right, that if a change is not inherited, then evolutionarily speaking, it has no meaning. It might be interesting, but it has no meaning if it doesn't go pass on to the offspring. Now, obviously, this is considered to be very, very important. However, one of the major insights of the last 20-30 years is that there are many other kinds of variations that are very important for evolution. So here I need to give you a quick terminology. So all of you have studied some version of the evolutionary theory when you were in schools or colleges. And I'm sure many of you are probably even teaching that to your students right now. So the canonical version of the evolutionary theory is what got synthesized in around 1950 to 1955. And this is what is known as the modern synthesis. This primarily takes Darwin's views, then marries them with what Gregor Mendel figured out about genetics, and then it builds upon the work of a lot of very prominent evolutionary biologists like Ronald Fisher, Sewell Wright, Dobzhansky, Haldane, and so on and so forth. So all their theories, all their thought processes get crystallized into what is known as modern synthesis, and that's the canonical version you see and read at school. However, one of the problems with the MS, well, I won't call it a problem, but one of the features of the MS is it gets crystallized in 1950 to 1955 before the massive amount of knowledge that was generated in molecular biology. I mean, remember 1953 is when the DNA structure is getting discovered. And obviously the molecular biology boom that happened was post 1953. So much of the information that has been generated over the last 30, 40 years on molecular biology actually doesn't feature directly in evolutionary theory. Therefore, the last 30, 40, last 20 years or so, lots of biologists have been saying that we need a new program, a new research paradigm in biology called the extended evolutionary synthesis. What is it? That should take the various pieces of information, various pieces of insights that has come from molecular biology, developmental biology, etc., over after the modern synthesis happened, and try to incorporate it into a bigger theory, a more extended theory of evolution. And hence, it is called extended evolutionary synthesis. So, as I said, one of the major goals of this so-called extended evolutionary synthesis program is to try and figure out how the other kind of variations, non-genetic or supragenetic variations, all of them end up affecting the evolutionary process and therefore end up affecting what we understand about biology. So one of these things that we have now understood is about stochasticity, stochastic variation in gene expression itself. Okay, so for example, there was this paper in 2002. It's a very famous paper. I'm sure most of you would have come across it, where they were trying to see that if everything else is the same, then just by chance, just by noise, do I get a variation in terms of gene expression and therefore the phenotype. So what they did was they took strains of E. coli and then they took two different genes, fluorescence genes, the cyan fluorescence protein and the yellow fluorescence protein, and then they put them in the same chromosome under the same promoter. This is very important. I mean, you know that different promoters can act different. And you know the bacterial gene is, bacterial chromosome is circular. So you have the promoter, common promoter in the middle, and they put the two genes equidistant from that common promoter on the opposing sides of the origin of replicate. Now, what is happening over here? If all else is equal, if you don't have any noise in the system, then you expect that the same protein, say, will get expressed in similar amounts. And when you see the whole thing under a fluorescence microscope, you are going to see the same color. But in reality, what you see is all kinds of shades of color, meaning to say that the relative amount of the cyan fluorescence protein and the yellow fluorescence protein are varying across the cells. And as far as we can see, they are varying in a completely stochastic manner. There is no pattern. So which means that even if you end up having the same genes inside your body, then the pattern of gene expression might vary stochastically across cells. And obviously that itself can, in theory, end up having an effect on what is the phenotype that you are getting. So that was about genes. This is about environmental variation. So we know that even if you have the same gene, then depending on which environment you find the organism in, there can be serious differences in terms of the phenotype. So for example, what we are seeing over here is Daphnia. This is a small crustacean. And this Daphnia, it has several predators. One of them are these larvae called phantom midge larvae. Now these guys, they end up swallowing the Daphnia whole. Okay. Interestingly, if you simply take some water which contains this predator, and then you make the Daphnia eggs grow in that water, so you don't even need the predator per se, just make the Daphnia eggs grow in that water, then you will find that the Daphnia that is formed has some differences. One thing that happens is you can see this spine over here. This spine becomes elongated, and so this is the control one, which has grown in normal water. You can see that the one that is going in the predator laced water, it has longer spine. And can you see this stuff over here? Okay, so this is what is known as the helmet. And what it does is, when there is predator, the presence of predator, then it develops some teeth-like structure over here. As a result of that, now it becomes very difficult for the predator to swallow this. Okay. Now remember, genetically these two are the same. It's just that the environment is changing their phenotype. A more drastic thing is found in these larvae. This is some organism, Nemoria arizonaria, caterpillars. And these guys, they grow on oak trees. And the oak trees, during the summer, when the larva grows, it eats the oak leaves and then it resembles a summer oak twig. However, the larvae that are coming out in the spring, this is the time when the when the oak tree is flowering, at that point, they have a very, very different phenotype. This is the larva. And if you look at it, this is resembling the oak catkins, the oak inflorescence during the springtime. So basically, depending on which part of the year they are growing and depending on what is it that they are feeding on, they end up changing their phenotype rather drastically. So these are classic examples of environment affecting phenotype. And classically, environmental variation was supposed to be non-important. It was supposed to be just a nuisance in studying genetic variation. And a lot of experimental design efforts were actually put in just to make sure that environmental variation is either not there or is experimentally controlled for, statistically controlled. However, one of the major insights that we have had over the last several generations is that environmentally induced variations can actually pass across generations. And when that happens, several very, very interesting phenomena can take place. And we are going to discuss some of those phenomena today. So a major conceptual stuff that one needs to keep in mind here is that selection deals not with genotypes, but it deals with phenotypes. Okay, what the organism's actual properties are. And this phenotype is a dynamic thing. Okay, reaction norm refers to what phenotype you get in what environment for a given genotype. And it is these dynamic properties which are the sole criteria of fitness in the struggle for existence, which essentially means that genotypic information is important, but what actually matters to evolution is what the phenotype looks like. And this is something that, I mean, there is one more stuff over here, which is the fact that there can be an interaction between the genes and the environment. So here is a very classic example of that from human beings. So we know that we have this neuropeptide in the brain called serotonin. And serotonin is something that is very, very important for maintaining the mood for people, right? Which is why when people get into mental health issues, one of the first things that the doctors prescribe is serotonin uptake inhibitors. Okay, so the idea is that serotonin is naturally synthesized in the brain, but if for some reason the serotonin level drops, then people get into trouble. So in order to prevent that drop, they give all these serotonin uptake inhibitors. Now, the way serotonin gets degraded in the body is because of the product of this serotonin transporter gene called 5-HT. Now, turns out that this gene, there are two alleles of it, call them capital S and call them S and L. So if you have two alleles, you have three genotypes, SS, SL, and LL. these two alleles, as far as we can make out, the protein that they are creating is identical. Okay, there is no difference at the coding level. However, there is a difference in terms of how much of that serotonin transporter protein are they producing. And turns out that this SS genotype produces very little of it, the LL produces a lot of it, and SL produces a mid-level. Now, if you look at the probability of people having these genotypes developing depressive episodes as a function of what kind of maltreatment they got in their childhood, you can see that if you are a LL genotype, it is hardly affected. If you are a SL genotype, it's moderately affected. But if you are a SS genotype, then it's greatly affected, which means if you are a SS genotype, then if childhood maltreatment can seriously, more than double the probability of your becoming depressive if you end up getting bad treatment during your childhood. Now, this is very interesting. But then the question is that, again, this is not going to be important for evolution if it doesn't pass to the next generation, if it is not inherited. So can these kind of genotype by environment interactions themselves, can they be selected for? And turns out that the answer is yes. So this is one of the classic studies on tobacco hornworms by Suzuki and Mizuth. So these hornworm larvae, typically these are green in color. However, sometimes you also end up finding a black mutant. Okay. Now turns out that these black mutants, their color is actually sensitive to temperature. So if you take these mutants, and while they are eggs, and if you end up shocking them with a small amount of heat stress, then some of them, not all, some of them end up changing their color. So this, what you have over here on the x-axis are 4 different phenotypes that are formed, and you can see that they are differing in terms of the greenness of the skin color. So they are given 4 scores: 0, 1, 2, 3, and 0, 1, 2, and 3. So if you shock them with heat stress at some point, then some fraction of them becomes score from 0, which is black, they become 1, another fraction becomes 2, another fraction becomes 3. Now, what they wanted to see is, since different guys are becoming, changing their color to different extent, can you select for this? So what they did was they created 3 different selection lines: selection for high plasticity, selection for low plasticity, and control. So what did they do? So every generation, they shocked the larvae, and then they had 3 lines: one in which they shocked but then allowed everybody to reproduce, one in which they shocked but they only allowed the ones which are forming the green color, so high scores, score 2, score 3, to reproduce, and over generation the average score of these guys went up. In the other one, they only took the ones that did not change their color and remained black, and over generation the average score of these lines went down. So these are the 3 lines: high plasticity, unselected, and low plasticity. And after some 13-14 generations, they took these larvae, they shocked them at different temperatures, and they noted what is the fraction of the larva that is changing the, and they ended up noting what is the average color score of the larvae. And what they found was, as expected, if you look at the low plasticity line, it doesn't really matter how much you shock them. They are going to remain black. They're not going to do anything. If you take the unselected line, then for low temperatures, 20 to 30, not much will happen. For higher temperatures, their score is slightly going to increase. They're going to become slightly greener. But if you take the high plasticity line, these are the most important ones. Under low temperatures, their score is going to be, they're going to be primarily blackish. But as you keep on increasing the temperature, they are going to become greener and greener. In other words, they have retained the ability to change based on the temperature, to change their skin color based on the temperature. So why is this important? This is important because classically it was thought that it's only genetically coded characters which are important. Then people showed that even environmentally induced characters can be important, and I'm going to give you lots of examples of that. And then here what we are showing is that even when you have genotype by environment interactions, even those can become very important in the context of evolution. Now, this has a big effect in terms of how we think populations are going to respond to things like climate change. Why? Because as you know that over the last 5-6 decades or so, the rate at which the rate at which the environment is changing is extremely drastic. Average temperature is going up a lot. The amount of environmental variation in pretty much all the climatic factors that you want to check, rainfall, humidity, etc., all of them, the variation has increased quite a bit. Now, organisms just have not evolved to take care of that much of variation. Okay, genetic changes are slow. Why? Because genetic changes, if they are coming from mutation, then they are dependent on the mutation rates. And as we are going to talk a lot about mutation rates today, mutations in general are a slow process and they are a stochastic process. So it is thought that most organisms which do not have the ability to relatively quickly respond to environmental changes across generation times, they are going to be at more severe extinction risk compared to those which have a more plastic response. Now, this obviously means that if you are in a variable environment, you are benefiting with greater levels of plasticity. If you are in a constant environment, you do not require that much of phenotypic plasticity. But then, does that necessarily mean that species, if they live for a long enough time in constant environment, are they going to lose phenotypic plasticity? Alternatively, phenotypic plasticity means that there is no change at the genetic level. So which means when environment is changing, they will end up changing their response. So if you talk about evolution defined as genetic change, then does phenotypic plasticity aid in evolution, or does phenotypic plasticity hamper evolution? Okay. Now, I know some of you are working in cancer, etc. So what do you think? I mean, phenotypic plasticity is something that is very, very important across many, many areas of biology. So what is your take on this? Does phenotypic plasticity aid in evolution or does it hamper it? I'll quickly stop sharing my screen for a minute. Anyone?
Yes, sir.
No. Anybody? I mean, anyone from the audience? Not you, Prem. No, this is seriously important. The reason I'm asking this is, see, from a evolutionary biology perspective, I always think of phenotypic plasticity as being very important. But most of you are not evolutionary biologists. Most of you are, coming from a very cell molecular biology background. So what is your take on this? Is phenotypic plasticity important for evolution or not?
I think so. It is important because whenever it is communicated genetically to the next generation, it will add to variation and it will add to evolution in turn.
It will add to the plasticity part of it. Okay. But think about it. If there is plasticity, then it doesn't require any change at the genetic level. correct? So you are equally adept at becoming good and bad. Okay, just let me give you some example where this becomes an issue. So you know that in India, there is a lot of agricultural research that is happening. People are trying to develop very good quality, both animals and plants.
Professor Dey,
Yeah, tell me.
just 1 minute. It's my humble request to all the participants. Actually, we want the session should be an interactive way. Otherwise, Professor Dey is talking and you are listening. That because under the current pandemic situation, it is very difficult to see you. So I am very happy that Professor Dey requested the participant to be as a panelist so that he can interact. I think, Professor Dey, am I right?
Yeah. I mean, that's the whole reason for which I wanted it this way rather than the conventional webinar.
Yeah. So that is otherwise like Rabindranath Tagore's drama Raja, where the all are behind and the person is saying, that is becoming useless, and sometimes it will be boredom. So as Professor Dey is asking, I think if you, the participant, personally and eagerly participate in an interactive way, you can extract more from Professor Dey. So, and other panelists. So it's my humble suggestion, humble request to you all. This is not a one kind show, one hand show. This will be basically a interactive way. Kindly do in such a way so that you, at least the panelists will talk 70 to 80% time and you talk 20 to 30% time, so that we can understand where you are and what we are. Okay.
See, there is one more reason for which it will be useful, if people talk. And that is that all of you have your own research program, teaching program, whatever. Okay. And it's very difficult for you to probably go and talk to people from a slightly different research program. And you will realize if you talk with each other and if you have a lot of interaction, that different people from different areas view the same problem very differently. So if you really wish to expand on how you think about your problems, it's very important that you say what you think. Otherwise, how will the other person know? And if you don't talk, then the other person will not talk. And I have nothing. I mean, I am a professional teacher. I can easily talk for 2 hours and another 2 hours without anybody saying a word. All right. So yeah, it will be a nicer thing if we have an interaction. And it will also be nicer if people who can afford in terms of their data and all can turn on their videos, because then at least one can see each other. And I think a discussion is much better if you can see each other. Okay. So, okay. Doesn't look like people are convinced about it. So fine, no problems. Okay. What I was saying was that in India, you have a lot of agricultural research happening. And one of the issues of doing agricultural research in India is that the climate situation in this country varies greatly from north to south, east to west. Okay. So you are, let's say, sitting in, many of you are in Kollani, right? So many of you are aware what is Vidhan Chandra Krishi Vidyalaya. So if you are sitting in Kollani and you have developed, let's say, a kind of rice, it works perfectly fine when it is in your place. You take that stuff and you now transplant it in Punjab and you transplant it in Andhra Pradesh, and you suddenly see that that very thing is not at all working. Why? Because the environment is different. So that is phenotypic plasticity for you. Correct. So phenotypic plasticity can actually mean that your evolution that you thought that you have, either by artificial breeding or by selective breeding, whatever, the variety that you have created, that's actually not having any effect. So in that sense, evolution has failed. Right. So phenotypic plasticity cuts both ways. it can be very good if the environment is what you want it to be. If the environment changes, then it can become very bad for you, isn't it? That is why I asked the question in the context of what you are studying. And the reason I asked it is somebody said cancer. What do you think in your context? Is phenotypic plasticity good or bad? Is it something that will be helpful or non-helpful? Most people in cell wall bio don't even think in terms of phenotypic plasticity. Most people in ecology evolution only think in terms of phenotypic plasticity. Okay, and that's why I'm just flagging this question. If you need more time to think about it, fine, no problems. But please appreciate and ask this question in the context of whatever you are doing. Is phenotypic plasticity a problem, or is phenotypic plasticity something that I really want in the context of whatever problem that I'm looking at? Now, obviously, if you're only doing bioinformatics, then you probably don't need to ask that question, but everybody else probably needs to do.
Hello, sir. I want to. Hello, sir. Myself Milul Maiti.
Milan Maiti.
Okay. So, one question: is phenotypic plasticity possible without change the genotype?
Phenotypic plasticity produced without the change of phenotypic plasticity, by definition, is when you have the same genotype giving different phenotypes in different environments.
Only environmental effect is there.
Yes, so the same genotype gives different phenotypes in different environments. So by definition, the genotype is the same.
Okay.
Okay. Fine. So I will go back to my PPT. Yeah. Where was I? Yeah. Okay. So this, what I talked about was in terms of what is the statistical nature of the variation. Okay. Now you can also think of variation in terms of the magnitude of the effect that it produces. Okay. Now this is something that is conceptually very interesting. Practically speaking, it may or may not be that simple to do it. Now, one kind of variation is what is known as continuous small effect variation, where you essentially have a trait, and what the alleles do, what the mutations do, is you get slight deviation from the trait. So a guy who is 6 feet tall, his kids are 6 foot 1 inch tall or 5 foot 9 inch tall. Okay, so small differences. These are typically continuous differences that happen. The other kind is where you get large effect variation. Okay, where let's say a completely new trait ends up coming, or from a pygmy like situation you suddenly become a Maasai like situation, and that is what can potentially lead to new functions. That is what can potentially lead to reproductive isolations, and this is what can lead to directly or through subsequent changes lead to formation of new species. Now, throughout the history of biology, okay, I'll take that statement back. When people started talking about mutations, when Hugo de Vries, for example, was talking about mutation, this is what he was primarily talking about. But then when people concentrated on the modern synthesis, the early days, the 1930s, 40s, 50s, that's when people completely moved their, focus on these kind of variations. It was thought that these are the ones that are important for evolution. If you look at molecular biologists are always trying to develop screens, right? They're always trying to figure out if I have a phenotype level difference or not. And then they actually ideally like these kind of mutations. But when they then couch it in terms of evolution by constructing those phylogenetic trees, they quickly shift back to this kind of variation, continuous small effect variations. Okay. So we have a very peculiar relationship between these two kinds of variations. Both of them are studied. This is the one that is studied more in the context of small bio, cell bio. This is the one that evolutionary biologists think about. And whatever theory we have about mutations primarily pertains to this. We have very little or no theory related to this, except for very vague statements like, oh, maybe things are changing at the promoter level for these kind of things to happen. Except for that, we don't really have a good appreciation of what leads to macromutations and how their effects are going to work. So if we classify variation on how it is generated, then there are all kinds of things. So for example, at the level of the chromosomes, we can have all kinds of changes, inversions, etc., etc. Then of course we have this huge, humongous thing called mutations. Okay, so many kinds of changes. This is something that I'm going to talk separately. Okay, because it deserves its own separate treatment. I'm going to talk about all these other kinds of changes, not all of them, maybe just first 2 or 3 of them, and then leave you with some amount of generic insights that we have about the nature of variation in biological systems. And of course, we have this question that does this even matter? For every one of these things, I'm going to ask this question: does this kind of variation even matter in the context of evolution? And if it does, then to what degree? Okay. So I think it's around 11:55. So we'll take a quick break over here for, let's say, 5 to 10 minutes. And then after that break, we'll come back and we will deal with these various aspects. Okay. So,
okay, sir.
Yeah, I'll be here if anybody has any. Chalo, somebody at least responded to the break part. Thank you. So,
Yes.
So, yeah, I'll be sitting here, and yeah, feel free to come back in next 5-10 minutes. And if you have any questions, feel free to ask them. I'm right here.
Dear participants, this is an interactive session as sir told. So you may unmute your mic and you can ask any questions, and you may directly interact with sir. And sir has told that we will be having a few 2 minutes break. And if you have to go for break, then please come back within 2 to 3 minutes.
No, I said 5 to 10 minutes, yeah, not 2 to 3.
Achha, sir, 5 minutes.
2 to 3 mein kya
So
hoga?
usually 2 minutes goes to 5 minutes.
I know. It's okay. See,
I have one question. This is Dr. Somuhaji. I am not actually a biologist. I am from a materials engineering background. So I want to know, does this evolution have any effect on genetic factors?
Classically, evolution is defined when the genetic factors are changed.
Okay.
Okay. So the answer is yes by definition.
Okay, that means it is some change in the cell structure and all these things also.
No. So evolution is when there has been change at the genetic level. Now the genetic change may or may not reflect at the cell level.
Okay,
it can reflect at so many levels. Maybe all your cells are the same, but behaviorally you have become something different. That will be construed as evolution.
Achha. So that way, since people, that several metallurgists are working in biomaterials development. Yes. Suppose we are designing any tissue, okay, by means of 3D printing. So we should be aware of this evolution and genetics, both way, vice and versa.
The point is, if you are generating a tissue to your own design, then there is no reproduction happening, right? Because you are creating the material, and suppose tomorrow I say, give me 1 kg of that tissue, you will do something in your lab and give it to me, correct? There is no self-propagation happening. So if there is no self-propagation happening, then you don't really need to care about anything. You just do it whatever way. However, if you are trying to draw your inspiration from a biological system, then it's a different ballgame altogether. Because, see, the point is, when suppose you are making a tissue, okay, you have a few characters in your mind, tensile strength, maybe, or I don't know,
corrosive compatibility and all these things.
Corrosive compatibility, yeah, whatever, depending on your usage, correct? You don't really care about any other property of the cell, correct?
Okay.
At the same time, when the organism is out there in nature, then the organism cannot function like that. Then the organism has to make sure that it is reproducing, right? Because if it is not reproducing, then it will not be able to continue over time. It will go extinct. So the constraints that a natural organism faces are very different from the constraints that are faced by any system in the laboratory. Okay. And that is why you per se don't need to think about how evolution got the organism there. You can just use it as an input-output system. Is that making sense?
Okay, I am getting certain points. Okay. Okay. Thank you.
Sir, I'm Korbi Ganguly. I have a question. Sir, continuous and discrete variation, are they taking place simultaneously or either or?
They can be simultaneous.
Then it is happening, when it is happening simultaneously, it is triggering the effect, or that is having two different line of effect?
See, these are two different kinds of stuff. Okay. For example, when I say continuous, you remember that skin color thing that I showed you?
Yes.
Okay. You have all shades. You have a very dark skin guy, you have a very light skin guy, and you have all shades in between. Okay. That's what I mean by continuous.
Okay.
Discrete, there can be two types of discrete. One is discrete in the context of the same trait. And in that stuff over here, if everybody else disappears and you only have the very dark colored guy and the very fair skinned guy, okay, and one mutation in the dark makes you very fair, or one mutation in the fair makes you very dark, that's a discrete jump. Okay.
So can we set the example of temperature dependent coloration in caterpillar green and black in this discrete?
Actually not, because what
that is environmental plasticity.
No. See, there are two different things altogether. Okay.
I
am taking variation and I'm just showing you in how many ways you can look at it. It's the same variation. So each way of looking at it, it's not mutually exclusive from the other. Okay. So this is like saying what kind of identities a human being has. I have identity as a Bengali. I have identity as an Indian. I have identity as a male. I have identity as whatever, lover of biryani or something.
Okay.
Now, all these identities are simultaneously existing, right? Similarly, all those different ways of classifying that I told you, they're simultaneously existing. Okay. So the first thing that I said, the genotypic, environmental, and the genotypic / genotypic cross environmental, that's a statistical way of decomposing variation. Okay. This thing that I said about continuous versus discrete, that is a different way of looking at variation. That is in terms of whether there are intermediate forms or not. Then the micro versus macro that I'm talking about, that is whether a novel feature is being generated or a novel feature is not being generated.
Okay.
Okay. So these are all complementary ways of looking at variation.
Okay. Thank you. Thank you, sir.
Yeah. Okay. So let's give a few more minutes.
Good afternoon, sir. My name is Niranjana Bhattacharya Nath. Yes, sir. Actually, you have mentioned a few points. Those they are very important, and those they basically directly or indirectly regulate the overall variation. For example, you have mentioned mutation, recombination, epigenetic factors, and we all know these are very much important. So my question is, sir, which source among these anyone is most important to evolution? Is there anything like that, or all they are having the equal importance?
Yeah, that is exactly what we are going to discuss, because I just showed you a few names. Now I'm going to take each one of those names and I'm going to show you examples of that, and thereby discuss when it becomes important and when it is not important.
Okay, sir. Okay, sir. Thank you so much, sir.
Yeah, that is the next 1 hour. Okay, so I think we can probably start now. It's about 12:05. Where am I? Yeah. Okay, can everybody see the slides?
Yes, sir, it is visible.
Okay. So as I said that mutation is going to be a separate talk altogether. I'm going to talk about the other things. Recombination. Okay. Now, as we all know, recombination is something that is happening only in sexual organisms. So since there are non-biology people over here, what is recombination? So during, sexual organisms, you know that one cell from the male and the one cell from the female has to come together and fuse. Now, when that happens, obviously chromosome numbers will double. Therefore, before that happens, during the formation of the gametes, the chromosome numbers are fast halved. Okay. And during that process, which is called meiosis, a very peculiar thing happens, which is chromosomes, they exchange genetic pieces with each other. Okay. Very technically speaking, homologous chromosomes exchange genetic pieces. And this process is what is known as recombination. So this happens only in those organisms which are sexual, which means only in those organisms where you have a separate male and a separate female. Okay. Now, the reason it is important is because it puts the genetic parts of chromosomes in different combinations inside the gametes, and therefore it creates a new level of variation altogether. Unfortunately, in the same process, it also ends up exposing alleles to selection, because in a beneficial combination it's fine, but next generation, when again recombination happens, that combination breaks, and therefore it also can expose alleles to selection and thereby reduce variation. So this is a double-edged sword. It both increases variation of one type, recombinational variation, and decreases variation of another type, the genetic variation. Now, that's one type. The other kind of variation, what we have been talking in the context of phenotypic plasticity, is what is known as environmentally induced variation. And this became prominent with the work of Waddington in 1953. Happens to be a very important year in biology, I guess. So I'll quickly walk you through what Waddington did. So if you look at flies, Drosophila, okay, common fruit flies, then these are wings of these flies, and these wings, there is something known as a cross vein. So you can see that most of these are going horizontally like this, but there are two, one over here and one over here, which are going vertical, and they are connecting two major horizontal veins. So these vertical veins are what are known as cross veins. And what Waddington saw was that if you take the Drosophila pupae and you subject them to a quick heat shock for 2 hours, then sometimes these cross veins are broken. Okay, so this is what is known as cross veinless. So as far as Waddington could see, there was no difference in any other characteristics of the flies. They were equally fecund, they were producing same number of babies, they lived roughly the same amount of time and so on, except that there was this small, environmentally induced variation that happened. So what Waddington did was, he wanted to see whether this kind of a small little change, whether that can be selected or not. So what he did was, every generation, he shocked these flies, and in one set of lines, he took the ones which are cross-veinless and he allowed them to reproduce. And in another set, he took the ones which remained normal, which did not respond to the heat shock, and he ended up propagating them across time. And each generation he noted what is the fraction of crossbane-less flies, these guys, and what is the fraction of the natural flies, the normal flies. And as expected, what he saw was that over time, the frequency of the crossbane-less flies is going up in these, so that by about 17, 18 generations, the entire population was crossbane-less. And in this, okay, it didn't go to 0, but it was about 80% of the flies had crossbanes. Very interesting. Now comes the important part. Once the whole thing reached somewhere over here, Waddington showed that even if you don't heat shock them anymore, even then they are producing crossbane-less flies. Now this is very, very critical. This experiment is one of the classic experiments in biology. Why? Because you remember the tail cutting experiment of Weismann. Okay, what had he done? He had taken mice and he had every generation cut their tails, and he had shown that even after 19-20 generations, the baby mice that were being produced in the next generation, they still had their tails. Okay. Human beings have been doing this experiment for, I don't know, last 2000 years in the context of Jews, and last 1400 or so years in the context of Muslim males who undergo circumcision. And yet every generation, the circumcision thing has to be done anew, simply because acquired traits like these are not inherited. Okay. However, what Conrad Waddington showed over here was that an acquired trait of crossbane-lessness is actually getting inherited, because once they have reached this point, even if you don't heat shock them, they will still keep on producing crossbane-less flies. This led to a very, this led to Conrad Waddington proposing the process of what he called as genetic assimilation. So what happens? So remember, we are talking about phenotypic plasticity. So this is phenotypic plasticity over here. You have a phenotype in a given environment. Now the environment shifts and the same genotype now shifts to a different phenotype. Okay. Now, if you keep on doing this generation after generation, after some time, even when you have brought it back to the old environment, the phenotype will not come back to the old environment. It will stay put in this state. And this is what Waddington called genetic assimilation, the process by which environmentally induced variation responds to selection and ultimately becomes stably inherited. Okay. Now, this was a very big news even in 1953, but people didn't really know what to do with it. Okay. Till, obviously there are many other stuff that happened. However, one major thing that came into picture was the work by this lady called Mary Jane West Eberhard in 2005, where she gave a very different theory of how evolution might work. So if you look at the classical picture of how evolution works, what is it? We are told that you have mutations. Their mutations or recombinations, they create variation. Selection operates on that variation, and therefore you go from one trait to another trait. Okay, this is the stuff on which all artificial selection works, all crop breeding programs work. And if you remember some time back, I think 2 or 3 years back, 2018, I think, the Nobel Prize in physiology went to somebody who actually took bacteria and essentially evolved them under different antibiotics to figure out what are the antibiotic resistance properties that are evolved, right? So all that stuff is coming in this framework where you say that evolution only occurs on pre-existing variation. Evolution does not create its own variation. However, what Mary Jane West Eberhard is saying is slightly different. What she is saying is that when a trait, the origin of a trait, this can obviously be due to mutation, but it can also be due to an environmental change which can cause the appearance of a developmental variant. It doesn't have to be a mutational variant. It's a developmental variant which can end up giving a novel trait, which can end up giving a novel state. Okay. Now, when that happens, then now please do remember that inside the organism, no trait exists by itself. Okay. So if, for example, I create a mutation which makes me 6 feet tall, then that change has to work with a bunch of other things in the body. My skeleton has to become that much longer. All the blood vessels have to become that much longer. All the nerve cells have to become that much longer, etc., etc. Only then the organism will be able to survive. Now, typically, when we think about evolution, if you are a molecular biologist, you don't take all these complications into picture. Why? Because you are either working with bacteria or you are working with a cell line. And in a cell line, the number of traits that need to be accommodated are very, very few. Okay. However, if you think in terms of a complex multicellular organism, you realize that any mutation is not only going to change the one trait that I am observing, until and unless that ends up talking to and modifying a bunch of other traits, the organism is simply going to die. So what you really need is what is known as phenotypic accommodation, whereby you need a rearrangement of various aspects of the phenotype to the new trait, and this is made possible because our developmental system is inherently extremely plastic. Okay. Now, once this has happened, then you have one organism or a few organisms which have that new trait. However, evolutionarily speaking, that still doesn't mean much, because then this thing has to go forward. It has to go over time across generations. And initially, this thing is going to be happening because of this phenotypic accommodation and not because of the genetic mutation. Okay. Now, this actually solves a very major problem in biology, which is, if a new mutant arises, how is it going to spread in the population? Typically, people say, oh, if the new mutation has greater fitness, then it will very easily spread in the population. But remember, in no population do you have selection operating alone. In every single population, you have selection operating in conjunction with genetic drift. And because selection and drift are going to operate together, anything which is in very, very low frequency is going to be affected more by drift. And therefore, the conditions under which a new mutation can spread in a population are actually rather limited. I mean, this is something that theoretically has been shown, empirically has been shown, and so forth. So this has always been some kind of a problem with evolutionary biologists, that how does a new trait or a new variant, what exactly will ensure that it spreads? What this says is that you don't really need to think along those lines. As long as you have the new variant, which is coming about through phenotypic accommodation, which is coming about through phenotypic accommodation, then that stuff will probably end up happening in a larger number of organisms, simply because the rate of phenotypic plasticity is far greater than the rate at which a new mutation will come about, and therefore the initial spread of the new variant is still going to be much easier. And this is the hypothetical part. He talks about something called genetic accommodation. What he says is that once these three things have happened, phenotypic plasticity followed by phenotypic accommodation, if this keeps on happening, then somewhere or the other, genetic mutations will take over. A genetic mutation in the correct direction will happen, and that will get selected for, and then that is going to spread. Now, frankly speaking, this part over here is something that is very, very, how do I say, hypothetical. we don't have too many great examples of how genetic accommodation actually happens. This is a hypothesized process. However, we have a lot that we have seen over here and here in this trait origin and in this phenotypic accommodation part. And how those things happen is what now I'm going to now discuss. And the first thing that comes about, obviously, is what is called epigenetic variation. Now, most of you obviously understand what is epigenetic variation. However, since there are one or two people who are not from bio background, I'll quickly mention what is epigenetic variation. So, most variation people used to think happens because there are changes at the genetic level. However, we now know that it is possible to have changes not at the genetic level, not encoded by the DNA sequence, and yet that can end up having changes at the phenotypic. So, for example, you can have methylation of DNA. So these are adding of methyl groups on the DNA, as a result of which the rate at which and the time at which these genes are going to be transcribed and thereby lead to the proteins, that ends up changing. So the expression level of the DNA gets changed. You can also have modifications at the level of the histone. So histones are proteins on which DNA is tightly bound to form chromosomes. Again, if you change the histones, then the way in which the DNA unwinds to form mRNA and thereby proteins, that ends up changing. So again, there is a rate change over there. Or you can have changes through what are known as microRNAs, small 17 to 25 nucleotide long pieces of RNA. And it has been shown that many of these microRNAs are themselves epigenetically regulated, through these processes. So there are several examples of epigenetic processes. So I'll just give you one or two. So for example, if you look at rats, and more specifically, if you look at rat moms, then you will see that rat moms have a particular behavior which is known as licking and grooming. So when the rats have pups, then in the initial part of their life, the rat moms end up licking them and it ends up pawing them like this. That's what is called grooming. Okay. Now, it turns out that there's a variation between the rat moms. There are some rat moms who do a lot of licking and grooming, and there are some other rat moms which don't do a lot of licking and grooming. They just do the bare minimum or less than that. Now, turns out that there is a huge difference, behavioral difference, in terms of what the pups do when they are raised by a high licking grooming mother versus what the pups do when they are raised by a low licking grooming mother. So pups raised by high licking grooming mothers, they grow up to be nice, relaxed adults. They won't be afraid. They, if you put them in a bad situation, they will try to get out of it and so on. Whereas if you take pups which are raised by low licking grooming mothers, then they grow up to be anxious adults. So if you try to, for example, you just hold them by their tail and you try to dip them in water, so obviously they will drown. So these these anxious adults raised by low licking grooming mothers, they won't even try to prevent that. Okay, so normal rats, they will curl up like this and they will try their level best to escape from that situation. But these anxious ones, they won't even try. They will happily allow you to dip them in water and basically commit suicide. So it has been shown, very nice, elegant experiments, that this happens not due to any genetic reasons. These happen due to epigenetic reasons. Now, what are the epigenetic reasons? So what I'm giving you is a highly simplified version. The actual story has a few other players, but roughly speaking, what happens is that there is this gene in the hippocampus, the brain of the rat, which is known as the glucocorticoid receptor gene. Okay. So when a rat baby is born, then the promoter, this is the sequence which leads to, to which things must bind. Ribosomes must bind before the proteins are, before the mRNA is created. So the promoter of the hippocampal gene is highly methylated, which means there are lots of methyl groups sticking to it. Now, when the rat moms, they do a lot of licking grooming, then this methylation signals, they are removed. When the methylation signals are removed, so I'll just give you some example of what I mean. So here, if you look at it, these are the low nurturing mothers, and you can see that close to 100% methylation of the promoter. These are the high nurturing mothers, and the methylation level of the promoter in their baby's brain is about 10% or so. Now, when there is a lot of nurturing, then this methylation is reduced. When the methylation is reduced, then the glucocorticoid receptor gene is expressed, and the number of glucocorticoid receptors in the brain of the baby goes up. Why is this important? This is important because when the baby grows up, okay, then whenever there is a situation that creates anxiety, you know that adrenaline, noradrenaline, corticosterols, all those things are created in the body. They are synthesized, right? Now they help in the fight to or flight response. However, if they stay in the body for a very long time, then that is harmful, because once the fight flight response is over, the need for it is over, you need to come back to a ground state. So normally these corticosterols, they are brought to the brain and they are reabsorbed inside through that glucocorticoid receptors, and they are degraded. So that's how the body comes back to its original state. However, when you have a low nurturing mother, that its baby is someone which is going to have very little glucocorticoid receptors in the brain, then these cortisols, they cannot be reabsorbed. If they cannot be reabsorbed, they stay in the body, and therefore the anxious state of the organism that continues. Okay, so this is the quick, brief reasoning for why is it that these guys as adults are anxious. Now, turns out this is all good and nice, but until and unless this is something that is inherited, it is not going to matter in an evolutionary sense. Okay. And as far as we can see, this particular thing about this glucocorticoid receptor in rats, nobody has ever shown that it is inherited. However, there are other situations in which people have shown transgenerational epigenetic inheritance. Now, this is a crucial part. Why? Because in the body of most organisms, I'm talking sexually reproducing organisms, when a gamete is formed, there is what is known as epigenetic reprogramming. What is that? Basically, there are some enzymes which end up removing every single epigenetic mark that is there on the chromosome. However, and because that happens, people thought that epigenetic changes are a one generation change. They do not go to the next generation. Okay. Because that happens, you have quote unquote, saying that inheritance is not Lamarckian. Acquired traits, these epigenetic marks are acquired things. They are not inherited. However, one major insight that we have over the last few years is transgenerational epigenetic inheritance is really possible. And the more we look for it, the more examples of it we are getting in the literature. So for example, this is one of the early examples of transgenerational epigenetic inheritance. If you take mice, pregnant females, and hit them with this chemical called vinclozolin, then that alters the methylation patterns. And then if you take these guys and if you look at their pups, the male pups, they have all kinds of abnormalities, testes, prostate, kidney, etc., etc. Now, if you take all the male progenies over here and check them for up to 4 generations, and up to 4 means these guys checked up to 4. I think there is a later paper in which they showed that it goes even further than that. It shows all of them end up showing the diseased phenotype, even if they have never experienced Menklozoid. So this was one of the earliest examples of transgenerational epigenetic inheritance. Another very classical textbook example is this flower called toad flax. So this flower, normally it exists in two forms. One is what is known as the forma typica, the typical form, where the flower is bilaterally symmetrical. And the other is what is known as forma pelorica, where the flower is radially symmetrical. Okay. And if you are a biology student, you should appreciate that going from one kind of symmetry to another kind of symmetry is huge, is very, very big change. Okay. So this form, the peloric form, was thought to be a mutant. However, when people in you can see this is 1999, when people actually ended up doing the gene sequences, they figured out that there was no difference at the genetic level. However, what they did find that one particular gene called the L6 gene was actually excessively methylated in the peloric mutant. And what they found to their utter surprise was that this was very stably inheritable. This was so stably inheritable that for all practical purposes, it looked like a mutation. When they studied a little bit more, what they found was something very cute. They found that there was a gradient in terms of how much methylation can happen. So for example, this is the typical form. This is this form, the pelorica form. So this is almost completely methylated. This is almost completely unmethylated. And they found 3 different levels of methylation arranged from low to high, going from left to right. And if you look at these spars, which are indicated by the arrows, you can clearly see that as you increase the amount of methylation, the length of the spars increase. And you can see how from the bilateral symmetry with increasing methylation through intermediate forms, they are going to the radial symmetry. Okay. So, great. But do we have some more examples? So this is a very classic example, in human beings, coming from Radke et al. 2011. So remember that glucocorticoid receptor gene. Now that analog of that gene also, can be found in humans. And what they did was they studied the methylation status of glucocorticoid receptor genes in mothers and their children at 10 to 19 years after birth. Okay. And they also looked at what they nicely said, intimate partner violence, which basically means whether the moms were being abused physically and mentally before, during, and after their pregnancy or not. And what they found was that the methylation status of the mother's glucocorticoid receptor gene was not affected by IPV at all. Okay. However, if you look at the offspring, their methylation genes, their glucocorticoid receptor genes, when the mother faced, whether the mother faced violence before pregnancy, there was no difference in the offspring. When the mother faced violence after pregnancy, there was no difference. But when the mother faced violence during pregnancy, in that condition, those guys where the mother faced violence, they had a much greater methylation level compared to those guys where the mother did not face violence, which essentially says that the methylation status of the GR gene of adolescent children is influenced by whether what is it that the mother is facing during the pregnancy. Now, this is simply showing that this will end up affecting something in the children. Okay. A much more stark example of this comes from Holocaust survivors. So people who were the adult offspring of Holocaust survivors, it was shown that they had greater risk for the development of post-traumatic stress disorder, depression, and anxiety. And interestingly, this was true even though in all the cases that they studied, the offspring were born much after their World War II. And in this particular case, they actually figured out that this is happening due to epigenetic modification of what is known as the FKBP5 gene, which, surprise, surprise, turns out to be a very important regulator of the GRG. So, in both the human cases that I showed that this can go to the parents, from the parents' generation to the offspring generation. But for it to be evolutionarily important, it's not enough to just go to the offspring generation. It has to go a lot further. Okay. So does it go a lot further? So there was this nice study in 2014 where they studied what are known as epigenetic, what are known as epi alleles. What are epi alleles? So these are alleles which have the same DNA sequence, but their DNA methylation patterns differ. And what these people did was they created populations of this Arabidopsis thaliana that had a large variation in terms of epi alleles for two characteristics, flowering time and primary root length. Okay. Now, why is it that they chose these things? So whatever examples I had shown you till this point, they are all changes happening in one particular gene. Okay. So essentially single gene traits. However, a major argument in evolution is that most of the important traits important for fitness, they are not single gene traits, they are multi-gene traits, quantitative traits. Okay. So things like height, things like strength, things like endurance, things like pathogen resistance, reproduction ability, all of them are multi-gene traits. So people wanted to, people have been saying that whatever you are showing us in the context of epigenetic stuffs, they are not important evolutionarily simply because they don't affect the kind of traits that are evolutionarily important. So here they ended up choosing two traits which are very important evolutionarily for plants, flowering time and the primary root length. Both of these are known multi-gene traits, and they artificially induced epi alleles, and they showed up to 8 generations that these epi alleles are being very stably inherited and they are affecting the phenotypic variation in the populations. More critically, this is why I like this study, they actually went back to a massive collection of Arabidopsis thaliana from natural populations and ended up showing that around 30% of these various epi alleles they had induced over here, around 30% of these are naturally found, which means that there is a high possibility that even under natural conditions, these are going to be very, very important. So this is one way in which environment induces variation, epigenetic variations. And as I said, they can be, at least some of them can be transmitted across generations. Another way in which environment can induce variations is directly changing things at the level of the genes. Okay. Environment is doing it, but it's the genes which are getting changed. So one very nice example of this, again, this is Arabidopsis thaliana, which have been treated with a drug called geldaramycin. And you can see that when you treat that, these are the, the calyx at the initial stages. You can see that this is the control. This is the one which doesn't have GDA. And you can see that there is a huge variation in terms of what kind of forms you can get when you treat different genotypes with geldaramycin. Okay, major phenotypic changes happen. The much bigger example of this is a very famous study in 1998 by Susan Lindquist and Rutherford. So in this, what they did was they took Drosophila and they subjected them to heat shock. And when they did that, they ended up finding that there were massive variations that are being unearthed. And these variations, these are happening at the genetic level. Okay. So these are being stably inherited, and they actually showed what are the variations happening. Now, this is something that led to Rutherford and Lindquist talking about the heat shock proteins, HSP90 and all that. And their hypothesis was that under normal circumstances, you have a lot of variation in the population which is not expressed. Why is it not expressed? Because there are these proteins, chaperonins, which end up taking these variations and they end up nullifying them. Okay. However, when you heat shock these, then these chaperonins, they are heat shock proteins, so they are supposed to, rescue the organism from heat shocks. So they end up going and they end up doing their heat shock business, and now all these variations, they get expressed, and that is why you are getting so much changes. Now, this was a very interesting story. Chaperonins were a very big thing in the early 2000s and so. However, this story has been later challenged. What they ended up showing was that in this particular case, probably it was not due to heat shock proteins. What was happening was that when you heat shocked them, then there were lots of transposons in the genome. Their activity increased. And when their activity increased, then you know what transposons do. They just went and sat all over the place in the genome. And whatever variation you are seeing is because of those transposon activities. Okay. So the variation in some sense was indeed generated, after the shock was given. However, the question over here that people have been asking is that, does this have an adaptive significance? I mean, all you are saying is that there is a nice control which controls variation, and when you heat shock them, then this control breaks down. So what you are getting probably is a lot of mess. Can this mess in any way affect the adaptive potential of the organisms? And this was something that was shown very nicely in cave fishes. So you know that there are several underwater caves, and many a times fishes which exist on the surface, they, for whatever reason, end up moving to these caves. And when they move to these caves are completely dark, and therefore many a times after some time they end up losing their eyes. So that's why a lot of cave fishes are actually blind. People have shown using phylogenetic methods that these have come from recent divergences from fishes that have been existing on the top of the stream. So they asked this question, that is this something that is happening due to a cryptic variation? What they did was, they first took fishes from this top of the stream in the lighted region, and one of the things that they had seen was that there is a difference in the chemical quality of the water that is found at the top and water that is found at the bottom. Specifically, water that is found inside the caves have low conductivity. So they asked, is this low conductivity, can that itself be a thing? So they took fishes from the top and they put them in normal water and they put them in low conductivity water, and they measured two properties of the eye, the size of the eye and the size of the orbit. And so this is, I don't remember which one is which one. I think this is eye problem. This is the eye size, this is the orbit size. And you can see that this is the control. And the moment you do something as simple as changing the conductance, you can see that the variation in the population has gone up in both cases. You can see this same thing if you treat these guys by something known as radicicol. So radicicol essentially has the same effect as putting them in low conductivity water. So this is the control, and you can see that the moment you treat them with radicicol, all kinds of variations are unearthed, in which you can see that the size of the eye is going down, and you can see by the time you come to this one, there is no eye. This is a blind fish. Okay, one generation. Now, once they showed that low conductivity and radicicol treatment leads to similar kind of stuff, then what they did was something fantastic. So these are the control surface fishes. They treated them with radicicol. So again, you can see that the variation has gone up. Now they took these fishes and they made them to mate with each other. So basically from F1, they created a F2, and that F2 is over here. The moment you do that, you can see that here there is a major decrease in the eye size. So from a mean from here, the mean has come down over here. Similarly for the orbit size, suggesting that whatever variation is being induced over here through radicicol is actually a inheritable variation. And this is something that in this particular case, in the context of the fishes, this can have a adaptive significance. So the phenomena by which phenotypic variation can be exposed to selection in response to an environmental cue, the phenomena by which you have genetic variation out there and that genetic variation is normally not expressed, but it comes out when needed, when there is environmental stress, that is what is known as evolutionary capacitance. Now, although typically people have shown environmental reasons for this, sometimes the cue can even be internal to the cell. It's still environmental, but it's internal environment. So for example, in yeasts, you have this protein called protein sub-35, which actually helps to terminate translation when they are it has reached the stop codon. The ribosome has reached the stop codon. Now this sub-35, it exists in two conformations, the PSI plus and the normal one. Now in this PSI plus conformation, the sub-35 actually aggregates and then it kind of precipitates inside the cell, and thereby it reduces the concentration of free protein. Therefore, when you have the sub-35 in this conformation, then the concentration of the protein goes down and therefore the stop codon becomes less effective and translation continues past the stop codon. Now, once you have this extra tail, then there can be very significant phenotypic changes. Very interestingly, when you have normal circumstances, favorable for growth circumstances, then the yeast is not affected by this PSI plus mutation. It tolerates it. However, when you give stress to the cell, then the PSI plus phenotype actually ends up exhibiting a lot of variation, some of which can be potentially adaptively important. So a major conclusion over here is that developmental systems are often robust in environmental perturbations. Sometimes they are not, and that's when you have interesting things happening. Okay, so this adaptive evolution, the question that we initially asked, how does it matter for evolution? So if you think about it, natural selection is a very problematic thing. On the one hand, natural selection increases your adaptedness to the environment by taking out those variations that are not needed. At the same time, it reduces the amount of variation that is present because that's how it is programmed to work. So one solution that organisms have come up with is this thing called evolutionary capacitance, whereby the variation is there, but the variation is not expressed under normal circumstances. Only when situation becomes abnormal, only when there is some kind of stress, only then a lot of variation comes out, and then natural selection operates on it and selects whatever variation is needed. So you have a, a buffer stock of these kind of variations, and how that ends up affecting evolution is something that people are still thinking about, still debating about. So, anybody wants to ask something? Okay, so I just take about 5-10 minutes more, and I'll quickly give you a few insights on variation. Okay, so although I talked a lot about variation, fundamentally speaking, certain features are conserved. So for example, all cellular organisms use DNA as their genetic material, no exceptions. Genetic code by and large is conserved, although there are a few rare variants. And most basic molecular cellular processes, DNA replication, transcription, translation, they are by and large similar across all species. Okay. So there is a fundamental core out there, and that fundamental core seems to be roughly this. And this suggests two things. Either there are very few ways in which one can make life work, or as people have pointed out, maybe this is just a historical contingency. This is how our last universal common ancestor looked like, and therefore we are just carrying forward that contingency. Or maybe it's a combination of both A and B. However, okay, this is not very critical in this context. One thing that we know is that the genomes of complex organisms contain a lot of junk DNA, and just because we do not know what their function is, it does not necessarily mean that none exists. And one of the major things that people are trying to do in biology today is to trying to figure out what is it that this seeming junk DNA is doing. And one of the suggestions that people have been making is that maybe new genes actually come from junk DNA. So typically people used to think that new genes come from gene duplication, but now more and more people are gravitating towards the possibility that new genes come from junk DNA, in which case that problem about origin of novelty in organisms is probably resolvable if you think in terms of, if this hypothesis or this notion works out. This, most of us know, gene numbers do not really predict organismal complexity. So you have something as simple as rice, which has 41,000 genes, whereas humans, which by any measure are infinitely more complicated, have lots of more traits. They have only about 20,000 genes. This is obviously analogous to C-value paradox, but then one can solve the C-value paradox if you think in terms of junk DNA. There are several possible solutions that people have suggested to explain this. Maybe it's due to alternative RNA splicing, which substantially increases the number of combinations that you can get from a limited number of genes. Maybe organismal complexity is depending more on number of molecular interactions than on the number of genes and proteins. Absolutely possible. So this is what proteomics people are trying to figure out. And maybe gene expression and regulation and post-translational modification themselves are another layer on which, another layer of variation, which can operate on top of whatever you have in terms of the number of genes. These are all questions that people are actively researching right now. As of this moment, we don't have sufficient data to compare or check out these hypotheses regularly. One major way by which people have been talking about variation happening is through horizontal gene transfer across species boundaries. It's very common in prokaryotes. It's very, very less common in eukaryotes. And there are three major ways in which can happen: transformation, conjugation, transduction. If you don't know what those mean, it doesn't really matter. Practically, this is very important. So for example, things like antibiotic resistance, it is thought that horizontal gene transfer is the primary reason for that. Conceptually, it changes the way we do phylogenetic trees, because we do phylogenetic trees. I mean, we conceptualize evolution as a branching process, and that's why this is what our phylogenetic trees look like. Whereas if you really think in terms of horizontal gene transfer, then you should have evolution as a reticulate process as opposed to a branching process, and your phylogenetic trees should look like these. So this led to people trying to figure out what fraction is of the variation is vertically transmitted from parent to offspring and what fraction is horizontally transmitted. So there was a massive study in 2007 where they looked at some 246,000 genes and ended up showing that there is a difference in terms of which genes or the propensity of genes for horizontal gene transfer. And they showed that genes related to translation are the most recalcitrant to HGT. However, you need a lot more data on other genes. And in order to figure out the relative importance in eukaryotes versus prokaryotes, there was this massive study in 2015 where basically what they ended up showing is that in eukaryotes, most of the transmission is vertical, very little horizontal. In prokaryotes, there is a massive, both horizontal and vertical play. I won't say equal role, but it plays a much, HGT plays a much bigger role in prokaryotes. This I already talked about. Recently, more and more evidences have started accumulating that de novo genes are coming from junk DNA. And if this turns out to be correct, then this is going to be the next big news in evolutionary biology. This is also going to be a very big news in a lot of protein engineering and other stuff people do, because people typically try to just take one gene from some existing organism and trying to put it over here. If you have a new gene made from the existing genetic material, the way it might function in the context of the body might be very different. Again, we still don't know. We are still trying to figure out. This is not very important. This one is very important. Intraspecific genetic polymorphism. Okay, how much variation is there within a species? We know that there is a lot. However, interestingly, most of it actually has been shown to be neutral. Okay, there is something known as conditional neutrality. I won't get into that, but if that works out, then this might be a completely new way, a new store of variation for evolution to work on. So that's pretty much it as far as variation is concerned. This was as I said, an overview on variation. And after lunch or 5 o'clock onwards, what I'll do is I will take one aspect of mutations. Okay, so what I told you right now is, as I said, more teaching related stuffs. This is more a overview of the thing. The latest work, etc., the latest thoughts, all those will come in the mutation talk, where I'm going to talk about our recent understanding about how mutation rate works and how that interacts with various features. Okay. So that's it from my side. If you have any questions, tell me. Otherwise, we can break for lunch.
Hello, sir. Good afternoon.
Good afternoon.
Myself, Shahana Sengupta. I have a question to you. So when you told about the methylation of GR gene, especially in case of the rats, you have taken the examples. I found it very interesting, sir. Thank you. And after that, you have also given the example of human during the pregnancy of woman. And that time also you have given some data regarding this. So in that context, my question is that, sir, if a woman is suffering from this kind of problem during the pregnancy, up to how long generation the next upcoming generation will face the problem regarding that faulty methylation in the GR gene? Sir, can you tell me something regarding that?
Quite honestly, we don't know. Okay. The stuffs that I showed you, if you look at those data, and if you look at from what time period that data is coming, you will realize that we still haven't got sufficient time for multiple generations.
Okay, sir.
Isn't it? So as of this moment, we don't really have much data. Frankly speaking, the two human data that I showed you, those are a little bit of a issue. Okay, why are they issue? Because if you think about it, you know that in humans you have the germline, right? Yes. And the germline is inside the mother. Yes. So when the mother is getting affected, there is a potential that the germline is getting directly affected. Okay. So when you are talking about transgenerational changes, then in principle, you actually need to look at after one more generation. You actually need to look at it in the grandkids.
Okay, sir.
Only then you can definitely say that it is happening transgeneration. Otherwise, there is a possibility that it is within that generation.
It may inherit or may not inherit. But for that, we require much more data analysis.
We need a longer time.
Yes, sir, yes. Yes.
This is, see, the problem with these things that these are very new. Okay, I mean, you know how new epigenetics is, right?
Yes,
so, therefore, we in humans, we seriously don't have the data. But if the humans behave like the rats, then there is a huge possibility that these will go for at least some of them will go for multiple generations.
Okay, sir. Okay, sir. Very interesting topic I found at that aspect. Thank you, sir.
Thank you.
Dear participants, this is the question answer session. So please participate in this session. If you have any doubt and any query, please do ask here.
See, if you are a teacher, Prem, then the first thing that you learn is 1 o'clock को query नहीं करते, 1 o'clock को lunch के लिए जाते हैं, because any query is just going to, separate the difference between you and the lunch. So yeah, I completely appreciate. No problems. We'll break. If you have any queries, then I will take them in the next session at 5 o'clock.
Yeah. Hello. Am I audible?
Professor Banerjee.
Yeah, absolutely. You are right. So do not be between the lunch and the, so no questions. So it's just perfectly 1 now. So our session is 11 to 1 and we are trying to keep just like a very punctual way. Starts at 11 and we will finish at here 1, and absolutely we're right. So do not delay our lunch because food is very important. And the second thing, what I am telling, I am requesting all the participants this is an interactive session. So please ask your questions. And one more thing I am requesting you after this, please join. I found actually 36 persons have joined, they registered, but I'm seeing that 32 persons are there. So I do not know where are the other 4 persons. So basically, I do not know it is whose loss, my loss of myself or yourself. That's the problem. That's a big question. But please be here by 4:55 so that we can start at 5. I think it's okay, Professor Day.
5 o'clock, yeah, perfect.
Yeah. So I am requesting all the participants, because if you are not here, if myself and Professor Day is there, we can chat over phone, we can chat over it. But if you are not here, then it will not be possible for to start. So I am requesting all of you to be kindly present at 4:55 so that we can start on 5. Okay. And in the meantime, you please think about if you have any question, or surely at the initiation of the second session, at the initial part of the second session, you ask the questions. Professor Day is here to accept this question and gladly answer this. So just, I think after this, we should leave all and we will resume on 4:55. Okay.